We'll go ahead and get started. I'm Rob Burgess, and I'm Chief Business Officer for Sino-Biological. And I just want to thank all the participants for joining us today for our next installment in our series titled Lock and Key Immunodetection Technologies. We are very honored to have Dr. Jeff Silva here as our speaker today, Chief Technology Officer from Adeptrix Corporation. And in just a minute, I will properly introduce Dr. Silva. Just one main housekeeping item here, if you will. I ask all the participants, if you have a question, please type that question into the chat box. And then at the end of the seminar, I will walk through those questions and verbally ask those questions of Dr. Silva and give him a chance to answer those. So we will do all Q&A at the end of the seminar. So with that, it's my pleasure and honor to introduce to you our guest speaker for today. His name is Dr. Jeffrey Silva. Dr. Silva got his master's and PhD in chemistry from Johns Hopkins University, studying natural product biosynthesis and secondary metabolism in fungi. After graduate school, he pursued his postdoc at Harvard Medical School, pursuing and studying prokaryotic signal transduction of the model two-component systems, FOBR and VAN-RS. After that, he transitioned to industry, working as a senior scientist at a startup company called Microbia, which is currently known as Ironwood Pharmaceuticals, which focuses on bioengineering, fungal secondary metabolism, and signal transduction machinery to overproduce pharmaceutically relevant natural products. After Microbia, he spent many years working at Water Corporation as a development scientist in the area of global proteomics, utilizing novel LC-MS methods. He later joined Cell Signaling Technology, where he served as the director of the Proteomics Service Group. Recently, he's joined Adeptrix Corporation back in 2017, and currently serves as the Chief Technology Officer for Adeptrix. His goal here is to introduce a new assay technology platform that combines single bead immunoaffinity capture with multi-mass spec for a wide range of targeted proteomic applications. So Dr. Silva, it's a pleasure and honor to have you as our speaker today. And I will now stop sharing and turn the screen over to you for your presentation.
Okay, so let's see. Okay, please confirm that you can see the screen.
Looks great.
Okay, so first of all, thank you very much for the invitation to join here and give you this presentation. I'll be talking about technology that we've developed at Adeptrix called BAMS. The title of the talk is High Resolution Immunoassay Platform for Multiplex Biomarker Screening. We've been developing the technology for about 5 years, and over the last year, we've been able to launch the technology platform and reagents into a wide range of laboratories, including biotech, pharma, and academia. The assay technology is called BAMS, as I said, and it's a microarray platform for multiplex immuno-MS detection of targeted proteins and biomolecules. Before talking about BAMS, I'd like to point out a few details to illustrate the inherent nature of protein heterogeneity and complexity, proteome complexity. In the human genome, there are approximately 20,000 genes, leading to approximately 100,000 transcripts that's directed through alternate splice variants as well as through various promoters. These transcripts are then translated to over 1 million proteins arising from a variety of different post-translational modifications that cells use to modulate protein function and activity. These modifications include phosphorylation, acetylation, methylation, and others, as well as in vivo derived cleavage products. This means that any single protein can exist in multiple forms or proteoforms within the cell at any time. To illustrate this point of protein heterogeneity, I'm showing you two proteins, tau and actin, generated from Phosphocyte Plus, an informatics platform that is available on cell signaling technology, developed by cell signaling technology. The x-axis on the protein for each of the proteins shows the linear sequence of amino acids from the N-terminus to the C-terminus, while the y-axis indicates the number of annotated references that Phosphocyte Plus curators register into the database. That is associated with each designated post-translational modification within the amino acid location. So when configuring any targeted assay for a given protein of interest, one needs to keep in mind the inherent nature of protein heterogeneity, along with the challenges associated with the chosen method for the targeted protein analysis. And some of these challenges include the ease of use of the assay, the throughput that the assay is capable of, the ability to be able to multiplex if you're interested in monitoring more than one protein, the specificity for that assay, whether or not the assay itself is cross-reactive, whether or not the assay is able to monitor different proteoforms, and how easily it is to customize assays on that platform if you plan to extend the assay to include other proteins of interest later. So what you see here is the BAMS assay. The assay itself is conducted on a microscope slide. Each BAMS assay reagent is a single bead coated with a single antibody. As illustrated in the top right panel, you see 3 colored beads with different antibodies on them. The figure shows a slide with the BAMS assay beads on the surface to illustrate that each bead captures a dedicated analyte and deposits that targeted analyte in a localized position on the slide. The bottom right panel shows you how the beads are arrayed on the slide using a silicone gasket that forms microwells for the BAMS assay beads. Only one bead, which is 400 micron in diameter, can occupy a single well, a single microwell, which is 500 microns in diameter. When the captured analytes are eluted and deposited within each of the occupied microwells, we're able to monitor each of those targets using a MALDI mass spectrometer. Hundreds of beads can be arrayed on the slide to be able to perform multiplex assay. The BAMS assay platform provides the ability to monitor proteoforms and multiple PTMs within the same assay. There's no secondary antibody needed for the detection of the analyte, since the targeted analyte is measured directly by the MALDI mass spectrometer. So I'll go through the workflow with you in the next set of slides. Before that, I'd just like to show you that the BAMS assay platform can be used for a variety of different proteomic applications, including bottom-up methods, which include monitoring digested peptides. Here we have an example of phospho-Creb on the top left, as well as top-down applications where we capture and monitor intact proteins. And here we have examples of a small protein like insulin, CRP at about 23 kilodaltons, and proteins as large as IgG, here shown in the bottom right. One of the things that's important to notice is that the MALDI MS spectrum for each of these targeted assays have little to no background binding. The BAMS assay is very specific and shows clean elution of the target analyte of interest. One of the other things that I'll point out is that in the lower 2 panels, you see IgG and CRP. As the analytes get larger, you'll often see multiple charge states. So even though you see those 3 peaks, those are all the same protein, just in the form of a single, double, or triple charge version of that protein. So here I'd like to review how we configure BAMS assays through an efficient workflow, which allows us to screen commercial antibodies and other affinity reagents to benchmark its performance in the BAMS assay workflow. The workflow here is outlined for a bottom-up application, but we also carry out validation for middle-down and top-down applications. For those reagents that satisfy our criteria for affinity reagent specificity, based on the MALDI results, we qualify them as a BAMS reagent for a range of different applications. Typically, we'll carry out the testing using a number of different proteases to explore the flanking sequence surrounding the epitope region, allowing us to monitor different regions of the protein with the same antibody. To the right, I have several examples of these BAMS assays that I'll point out. Here in the top right, you see captured analyte from a tryptic digest of MKN45 cells. The antibody that's used here is directed towards the dually phosphorylated MET site, phosphotyrosine 1234 and 1235. And what we recover is a mass of 1852, which is which corresponds to the tryptic peptide of MET. And the important thing to see here is that the specificity is high. There's little to no background binding, and what we're observing is direct measurement of the analyte. A few other examples include 4-EBP. This particular example is for total protein. And what we observe here are two masses, 13, 13, 14, 69, 0 and 1 miscleavage product. We also have an example for phospho-BAD at serine 75 and phosphobeta-catenin at serine 552. So we can configure these assays to monitor these different proteins, different sites, and combine them in a multiplex experiment if we wish to monitor a panel of different proteins or peptides within a particular cell line or tissue. For validation, for any given assay, we have enough material that we're able to capture and deposit that we can carry out a number of different experiments. What you see here are just 3 different examples labeled linear, reflector, and lift. Basically, linear refers to a low resolution mass measurement that's still able to give us a mass measurement where we can securely identify and validate the identity of that peptide. If we need a more accurate measurement, we can perform the experiment in reflector mode to get isotopic resolution. So we have a more accurate measurement of that tryptic peptide. And lastly, we can actually sequence that peptide to get sequence validation and to validate the targeting of that specific reagent. A lot of what I'm discussing here today is outlined in a recent article that we published in IJMS. The reference is here, and it's also available on the resource tab of our website. The BAMS workflow is illustrated here as a simple method to capture, elute, and measure the target analytes. The workflow combines immunoaffinity capture with MALDI-MS detection, with the MALDI-MS instrument acting as a scanner. It's an assay platform that is accessible to a wide audience. I've included a link to a 90-second video, a 90-second YouTube video on the bottom here, for those of you who would like to view that video at a later time. If you search for Adeptrix on YouTube, you'll find a short and long version of this video. The longer version is 10 minutes, but you can look at that video at your convenience. So to start, the method itself includes the ability to analyze a variety of different sample types. Here, I've outlined or showing that we've used this in tissues and liquid biopsy samples with mouse, cell cultures. We've done some applications for COVID, looking at the spike protein. The start of the method basically involves generating a protein lysate. If you were to carry out a top-down application, to do bottom-up application, you would then digest that to generate the peptides that you would enrich. This is followed by enrichment, where here we have a 4-plex assay. Replicate beads for each of the target assays are used. So in the image here, you see 12 beads in each of the wells. Those 12 beads basically incubate with the prepared sample to capture, concentrate the analyte for subsequent measurement. After the capture, there's a series of short washes that include roughly 3 serial washes, takes approximately 20 minutes, and this will reduce any non-specific binding that occurs on the beads. After washing the beads, are transferred onto a microscope slide that's configured with a microwell gasket and a sample chamber gasket. The sample chamber gasket divides the microscope slide into equal size sample chambers that you see here with the black gasket that's set on top of the orange microwell gasket, so that you can run multiple experiments on a single slide. The sample chamber gasket comes in a variety of different configurations to accommodate different size experiments. They include 1, 2, 4, 8, 16, and 64 chambers. Once the beads are transferred, they settle into the microwells, as you can see here in the image above. And then next, the captured analytes are eluted from each of the beads within each isolated microwell using a matrix sprayer. The matrix sprayer delivers an aerosol of elution buffer within the microwells to expose the antibody to the elution conditions, which includes low pH and the presence of organic solvent, along with the matrix itself that helps facilitate the MALDI measurement. The antibody is tethered to the bead, so the only material that is deposited onto the surface of the slide is the captured and released analyte. Once the slide is dry, the gasket can be removed and the beads are discarded, and the microarray slide can be measured by the MALDI instrument. Here we have a 50-plex assay with an overlay of all the MALDI MS spectra collected for each of the targeted analytes. For the 50-plex assay with 3 replicate beads per target, we have roughly 150 MALDI MS spectra overlaid in that bottom spectra. If we focus on the individual spectra highlighted in that lower panel, we see the replicate spectra for the 3 different analytes that were targeted in this assay. And here we can see that that each of the single MALDI MS spectra are reproducible with little to no background binding for each of the targeted assays. So up to this point, I've outlined how BAMS can be used as a simple workflow with throughput that's capable of hundreds of samples a day using a multiple chamber sample gasket. With the data that follows, I'll illustrate a number of different applications to highlight the high level of multiplexing, the specificity and versatility of the platform, as well as the quantitative nature of the assay. Throughout the development efforts, our development efforts at Adeptrix, we've accumulated BAMS reagents in a variety of different areas, including oncology, neurology, and epigenetics, and this is what I'll focus on. Here we have an example of a targeted assay in the area of neurology showing intact capture and detection of TTR, or transthyretin, from a recombinant protein in a background of BSA, which is the top spectrum. Here we see a single and double charge of this protein, double charge form of this protein. In the middle, we see, in the middle spectrum, we see a negative control with no signal at all. And in the bottom, we see retrieval of TTR from CSF. So the difference between the recombinant and the endogenous is that the recombinant is full length, including amino acids 1 to 147. And in CSF, we have a processing of CSF that removes the first 20 amino acids, leaving amino acids 21 to 147 that accounts for that shift in mass. This is an assay where we use the recombinant protein from Sinobiological as well as the antibody, monoclonal antibody, and we're able to configure this assay fairly quickly within about a week or so to validate the performance of this and use for BAMS assay. Here we have another example where we were able to obtain recombinant protein from Sinobiological as well as several antibodies. I'm showing you an example from one where we have capture of intact alpha-synuclein and a negative control, which is just bead alone with no antibody. showing specific recovery, little to no background binding, and as illustrated by the clean signal of the target masses. And again, this is a top-down application retrieving the protein of interest from the sample. We're continuing to develop this, and we will be looking at liquid biopsy samples to capture and monitor this particular protein in a variety of different sample types. To illustrate the versatility and customization of BAMS, I'm showing you how we've configured multiple targeted BAMS reagents to tau protein to be able to monitor tau at different locations within the length of the protein, from the N terminus to the C terminus. The reagents were configured to capture proteolytic peptides to tau, generated from standard bottom-up methods using proteases such as lyc, arg-C, trypsin, and asp-N. Here you can see an example of two reagents, one at the immediate N terminus and the second in the middle of the protein located in an area that's known to be highly phosphorylated. So in that first spectrum, what we see is the single and double charged form of that peptide at the immediate N terminus, containing amino acids 1 to 24. And in the second spectrum, what you see is a more complicated pattern. And this is because this region of tau is highly phosphorylated. And so those different peaks correspond to multiple forms of tau representing different phosphorylation status of tau in this sample. And the sample that we've used here is human brain. In this example, we configured an assay to phosphothreonine 231. What you see here are two spectra, one in the positive direction, normal, and the other in the negative direction, which is disease. The disease sample is Alzheimer's. If we focus first on the top spectrum, the assay itself was directed to phosphothreonine 231. You can see upon lyc digestion, you generate this peptide which has a mass of 1046, providing us with the ability to recover that peptide using the BAMS reagent. And you see that clean signal with that mass. In the disease state, what happens is you'll notice that adjacent to that lysine at the end, at the C terminus, there's a serine. That serine gets phosphorylated in the disease state. And so if we look at the bottom spectra, you can see that there's a serine lowercase in blue that is phosphorylated. When that site is phosphorylated, lyc will skip that site and not cleave there, and basically cleave at the next available lysine downstream. So in the disease state, when there is hyperphosphorylation in this region, we generate a larger fragment as well at 1683, producing a mixture of both singly and doubly phosphorylated tau, which is a measure of hyperphosphorylation of tau using this reagent. The reagent itself recognizes phosphothreonine 231. Both of these peptides have that site, and that's why we're able to capture and measure both forms using this particular bead. This is another example of a tau assay to a different region of tau that's known to be hyperphosphorylated. In normal state, we can see up to 4 phosphorylations. In Alzheimer's disease patient, we can see up to 8 phosphorylations. And so this, in and of itself, is a nice assay to monitor hyperphosphorylation of tau within this region. Here we have another assay dedicated to amyloid beta. In this particular assay, we're using an antibody that targets this upstream, this N-terminal region, DAEFR sequence up towards the N-terminus, that allows us to capture degradation products to amyloid beta. The antibody, the reagent allows us to access degradation products between amino acids 672 and 713. And so here we're on bead multiplexing, capturing the population of peptides that are present, This is a middle down application where there's no digestion. We're just recovering we're recovering in vivo degradation products from multiple samples. And this is an overlay of normal and disease samples. In this case, normal is red, disease is black. And if we look at the data, look across the two samples, we can normalize using a common peptide, and in this case, the one that's highlighted in blue, A-beta 140. By normalizing to A-beta 140, we can see separation of the normal and disease state, showing us separation of A-beta 1 to 42, as well as other degradation products, with some of the other degradation products showing no separation. This sample itself is, the sample that we used here was CSF. So for the next slide, I'm just going to review I'm sure many of you are aware of what an ELISA assay is, but I'm just going to describe an ELISA where we go in to monitor a particular site on a particular protein. And in this case, I'm going to model it after histone H3K9 acetylation. So the first step in this assay would be to immobilize an antibody that recognizes total histone H3. And then the result is that it will immobilize histone H3 that contains, a fraction of which contains the site of interest and a fraction that does not. We go in with a second antibody that recognizes that site of interest, in this case, K9 acetylation. And then we go in with a detection antibody to provide us with a relative intensity, showing us the presence of that modification in our sample. Looking down below, we can see a number of different permutations, knowing of the different types of post-translational modifications that can happen at the tail of histone H3. Knowing that histone H3 tail is highly modified, there could be a number of different proteoforms that are present within this ELISA signal. And so, given the ELISA assay, we're not able to tease apart these different proteoforms. And so what we did is we configured a BAMS assay to highlight how we're able to monitor these different proteoforms by targeting this same region. Again, and I'll illustrate that in the next slide. So in this example, what we've done is we've done a lyse digestion. So we're retrieving a fragment of the tail and recovering all the K9 acetylated peptides from that. And what you see are a series of 8 peptides that are labeled P1 through P8. Because each of these have discrete masses, we can separate them by mass by virtue of the MALDI-MS spectrum. If we normalize to a common peptide like P3, we can see a region that's very different between normal and disease. If I hone in that region, what you'll see are these two regions, P5 and P7, that contain not only the K9 acetylation, but there's methylation at K4, mono, di, and trimethylation, and we can see that there's a dramatic difference between normal and disease state. So this is a nice example of how BAMS can be used to provide a higher resolution or a high resolution assay, immunoassay for different types of proteins that have a variety of different post-translational modifications and allow you to monitor these different proteoforms. In addition, we've applied this assay platform to be able to monitor different isoforms. In this case, if we look at the table, which is to the right, we can see that the chymotryptic peptides for ERK1, ERK2 in this region of this TEY, phosphothreonine, phosphotyrosine region, are identical. The mass is identical between the two isoforms. However, if we were to digest with trypsin, we'd be able to hone in on that region, that TEY region, and be able to resolve ERK1 and ERK2 by virtue of their discrete masses. In this case, we've configured the assay, used an antibody that recognizes that common sequence, and by using trypsin, be able to separate those two different proteoforms by virtue of their unique mass and be able to monitor the duly phosphorylated ERK1, ERK2 independently. This is an example where we've used the BAMS assay to monitor a point mutation. In this particular example, we analyzed a cell line, HCT116. HCT116 happens to be heterozygous with a missense mutation at position 107. So that valine right there is the point mutant. So this has a copy of not only the wild type, RAG, but also has a copy of the mutation, RVG. So when using this reagent that recognizes the immediate the immediate C terminus, which is that FEMD sequence, we can capture both the wild type and mutant form and observe both in this particular sample. We've used this assay in another way to monitor as an application to do epitope mapping for antibodies. And in this case, you can see a cartoon where for a given protein that might have different regions here colored, indicated by the different colors. If we have antibodies that target those different regions, and we're able we would be able to distinguish them by the unique masses that we observe that are specific to those specific regions. Here's an example where we applied this to a series of antibodies to KRAS. Here we looked at 10 different antibodies to KRAS, and by virtue of the three distinct MALDI-MS patterns, we were able to segregate them into 3 bins that target 3 different areas of KRAS. I'm showing you an example of how we were able to reduce the sequence to the known epitope by virtue of the minimum sequence, common sequence that is determined by the observed masses. In this case, the NTKS sequence, that middle spectrum, pointing to the region of group 2. Adeptrix carries out, for those of you who are interested in the platform, we're able to customize assays. If there are assays that are not available on the website, this is something that we can do. You can check out the BAM store to look at the assays that we do have available. Along with the products, you'll see product sheets that describe the different regions of the protein that we're able to monitor. The validation efforts that we perform not only allow us to utilize that for MALDI applications, but these reagents can also be used for LC-MS applications as well. We recently introduced a tool on our website that allows people to inquire whether or not we can configure an assay to any given protein of interest. And it's just a matter of basically typing in either the accession number or the protein name and sliding the location to the appropriate site of interest, whether it be a PTM site or an unmodified region of the protein, and inquiring with us whether or not we can make this reagent. We'll use this as a way to prioritize our development. So I would encourage people to take a look at that and see whether or not we can design an assay to your favorite protein. This is just to illustrate that we've done some work published last year, presented at ASMS, to illustrate how we're developing software that can quickly interrogate the results and display this in a way that does not require you to be a mass spec expert, but can present the data in an easy to use or easy to read platform. And with that data, we can cluster, perform statistical analysis and quantification to compare different conditions, different samples, and use it as a screening tool. To end, I'll just say that we've utilized the platform in a variety of different applications. I've highlighted some of these applications in this presentation. Some of the ones that I didn't mention, which you can find on the website, we've applied it to the spike protein for virology. I've shown you some epigenetics work. We're applying it to the use of testing allergens in food products for allergen testing as well. And with that, I will close and address any questions that you have.
Thank you, Dr. Silva. That was an excellent presentation. We appreciate your time today. It's fascinating technology, the combination of bead technologies with mass specs, truly powerful. And with that, because we're limited on time, I'm going to jump right into the questions. We've got some great questions for you from the chat box and specifically from a number of our participants. I'll start with Sanjay Danthy's question. I know you mentioned a number of different sample types that are compatible with your system. Sanjay is asking specifically about serum. Can your system be utilized efficiently to measure and detect analytes in serum, and then more specifically, maybe to accurately diagnose disease? And maybe you mentioned this and we just missed it.
Yeah, so I was not able to include this work, but there is an example in a poster that we presented at ASMS where we did carry out an assay for a particular serum protein. We collaborated with a company called Cis-Kappa Technologies, where they make anti-peptide assays for LC-MS applications. We wanted to demonstrate the utility of this on the BAMST platform. And you can find that in that poster. But yes, we can use this as a way to capture analytes in serum. What it does, the attractive thing here is that it allows us to directly capture those analytes on the bead, and as opposed to in an LC-MS workflow, you may need to do a depletion prior to doing the analysis because of the abundance issues you have with those top 20 proteins in serum.
Great, thank you for that. Vina S asks, is your system primarily geared towards research, or can it be optimized and really implemented, I guess, more broadly for diagnostics? What's really your business model there?
So our business model initially is really just to get the reagents out there and have scientists work with these reagents. In terms of utilizing the platform for diagnostic assay, this is definitely something that we do think is possible. But I think it's best for us to be able to put these reagents in the hands of researchers to basically do that work and help sort of move it in that direction. The thing that is helpful there is that Bruker has an instrument that is already FDA regulated, used for bacterial typing. So there is an instrument that's already been approved for this type of in vitro diagnostic application.
Great, good deal. Very quick yes or no question from Jeremiah Morrissey. Are the beads magnetic?
Yes, thank you for asking that question. I should have highlighted that in the presentation. Yeah, so the beads themselves are 400 micron diameter. They're magnetic agarose beads. It allows us to manipulate the beads very easily in the workflow. So yes.
Great. And another good question from Sanjay Danthy, and this was also followed up with a similar question from Ji Gao. Can you quantify the antigens in the sample, or is it more of really a yes or no question for detection?
With just like with any other mass spec method, with the appropriate internal standards, this can be used as a relative quantitation tool by spiking in either a labeled or truncated version of the peptide target or the protein. You can use it as a way to do relative quantitation. Right.
Let's see. Ayel N asks, is there a charge on the surface of the bead?
With it being an agarose bead, it's more polar. It's not a charged bead. There is a charge, I guess, because of the protein that's immobilized on it. We immobilize the antibody on the bead, so there will be a charge attributed to the conjugation of that protein to the bead.
Right. So Bia Annis asked this question. about mutations, is it that you need to know something about the wild type versus the mutant versions that you're trying to detect, or is it more of an open-ended system where it's discovery based? I mean, how much knowledge is really needed about the mutant versions of the antigens?
Yeah, so that's we ended up finding that example by accident. We had just analyzed HCT116 and noticed that there was a second peak there where it shouldn't be. And after researching what the genotype of HCT116, we recognized that there was an explanation for that second peak because it had a missense mutation. What this demonstrated was that you don't necessarily have to design an antibody specific to the point mutation. You can design an antibody to flanking regions that are perhaps around hotspots of specific proteins to be able to monitor wild type and mutant forms of that protein. Mm-hmm. And so there's enough literature, enough information out there for any given protein really to identify sort of the common mutations in these proteins if you wanted to configure an assay to monitor them.
Great. And Halkano Garacha is asking again about COVID research. And I know you touched on this, but what do you think its impact really is on COVID-based diagnostics or research in general?
So we're exploring that now. We started with the spike protein because not only did we want to demonstrate that it could be used to capture and detect the spike protein, but the reason for the choice of the spike protein was because it provided us a mechanism to actually concentrate the virus on the bead. And since Bruker has an instrument that is used for bacterial typing by simply transferring an amount of a colony on the slide, this provides the ability to concentrated deposit viral samples on a solid surface for you to directly analyze viral particles. Mm-hmm. We also plan to explore capture of different viral proteins as well, because we can multiplex. This allows us to have a panel assay for different viral proteins as well.
Great. Sutini N asks, can you use formalin-fixed tissue to do a characterization on that?
So we have done some work where clients have sort of optimized their sample prep for formalin-fixed sample processing. So the answer is yes. Typically, the method that you'll need to incorporate is a reversible method where the dialdehyde or the modifications that happen as a result of the formalin fix is reversed so that the antibody has a chance to recognize the epitope that it's been designed for.
Mm-hmm. Okay. And Charlie asks, how does your method compare to other methods such as CISCAPA? CISCAPA?
Yes. So I had mentioned that before in that poster, and I believe the year was either 2018 or 2019. If you go to the website, we did collaborate with CISCAPA Technologies and demonstrated that we were able to generate the same forward reverse curves that were generated on LCMS, on the LCMS equivalent. So that gave us the confidence that this platform can be used for an LC-free method for those types of assays.
Great. I have several questions about blood grouping. This is from Dina. As agglutination is a reaction occurring for blood grouping, is there such an immunoassay to detect this using your methods?
So we've definitely been able to explore a number of different applications, and it's questions like this that sort of nudge us to include another application. We have not yet applied it to that specific application, but what I would encourage you to reach out to me and discuss it further to see if we can build out an assay for that.
Great. And just a general question from me. I'm sure you mentioned this and I missed it, but what's your lower limit of detection or sensitivity?
We have performed assays. I'll give an example where we had done configured an assay for food testing or allergen testing for milk. We configured the assay for beta-lactoglobulin, and we were able to get about 15 parts per billion detection limit. So in contrast, the ELISA assay for that same target was specified or spec'd out to about 3 parts per million limit of detection.
That's fantastic. Just another quick question from me, and then I'll get back to one or two others from the chat group. I'm trying to get a firm grasp on your protease methodology and your cleavage of the antigens and why you want to do that, like the advantages to that. And I understand that different lengths of antigens may be wild type versus diseased, but can you just in a couple of sentences re-summarize why it's so important to apply proteases to detect different lengths or versions or different fragments of the antigen?
Yeah, so the use of the different proteases basically allows us to carve out a cassette that contains the epitope. So, and by having the choice of different proteases, if there's an arginine, for example, if there's an arginine in the epitope sequence that's needed for binding, then we can't use trypsin or ARG-C. We could use lyc, however, if lyc is able to carve out outside of that region and retain the epitope region so that we can capture that peptide. And so that's why we explore different proteases, so that we can say for this particular region, for this particular assay, trypsin, ARG-C, lyc will work, and these are the masses that you would expect. And for another assay, it may just be that ASP-N and lyc is the ideal protease because it does preserve the epitope and allow you to capture that intact.
Great, thank you. That clears it up. And then one other question there from Anthony. He's asking about, I think, glycated and glycosylated types of antigens. I mean, are you able to detect, I guess, sugar moieties and those types of modifications on antigens as well?
Yeah, so the way that we're tackling this is we can, spike protein is glycosylated for as many as we know. And so what you can do is you can use glycosidases to trim off the glycoform so that you can reduce that mass and help localize where the glycosylation is. So for example, if we were to capture intact spike and treat that with a glycosidase, we would see, we would be able to predict what mass a particular peptide is with an asparagine that might have a glycnac on it, and then see if we observe that mass. That's indicative of the remnant, the remnant modification after glycosidase treatment.
Great. Thanks for clarifying that. And this is the last question here. It's a good question. It's from Osmane Traore. He is setting up a biobank, and he's actually asking about the stability of samples during their storage over long periods of time. Is that an issue with your system where you need really fresh samples, or will your system detect the antigens and the fragments after a sample's been stored for quite a while?
Yeah, so from the context of storing biological specimens, that's sort of its own sort of series of protocols. Ideally, what we like to do when we're preparing samples for proteomic methods is we'll flash freeze them and store them at -80. If we have peptide samples, what we'll do is we'll typically purify them as digested purified peptides, lyophilized and stored at -80, and they can store up to multiple years. I've looked at peptides over 5 years that are in that form that are perfectly stable, including post-translational modifications like phosphorylation.
It's good news for somebody setting up a biobank that wants to use your bead assays, I think. So. So that's great. We got to everybody's question. We had a huge turnout today. So with that, I'll just take a minute or so to wrap things up. First, let me thank all the participants. Like I said, a very large turnout and some excellent questions from the participants. If you have additional questions for Dr. Silva, I believe he left his contact info there on his last slide. And you can also reach out to us to get his contact information. And Dr. Silva, I want to specifically thank you for an excellent seminar. We appreciate you doing this for the Sino-Biological hosted webinar series. You're clearly onto something very exciting at Adeptrix, and just let us congratulate you on all your accomplishments and your colleagues and the company in general. It's amazing what y'all have accomplished. So thank you again. And then I want to thank my colleague, Dr. Jen Liu, who's our marketing manager at Sino-Biological, for organizing and executing this webinar. It was a big success as usual. And with that, I will sign off and bid everybody adieu. Thank you again.
Thank you all for your time.
Bye.
Take care.